Mitosis is a process when a parent cell divides into two identical daughter cells, important for reproduction, growth and development, and tissue renewal. Interphase, the longest phase, includes three subphases: the G1 phase, S phase, and G2 phase. During G1, the cell reviews the cellular environment and the cell size to ensure that the conditions are appropriate to support DNA replication. When the cell has reached the desired size and is in a supportive environment for DNA replication, it is ready to exit G1 and begin the next phase of interphase, called S phase. S phase, or synthesis, is the phase of the cell cycle when DNA and the chromosomes are replicated. This event is important to the cell cycle because replication allows for each cell created by cell division to have the same genetic makeup. Similar to G1, G2 is an intermediate phase, a time for the cell to ensure that it is ready to proceed in the cell cycle. Occurring between the end of DNA replication in S phase and the beginning of cell division in mitosis, G2 can be thought of as a checkpoint, where the cell ensures all the intracellular components have been duplicated properly. After interphase is complete, mitosis can begin. In mitosis, a eukaryotic cell separates chromosomes in its cell nucleus into two identical sets of daughter nuclei, which is important for cell growth and repair. Mitosis can be broken down into four subphases: prophase, metaphase, anaphase, and telophase, where cytokinesis occurs. The first phase of mitosis is prophase. In prophase, the chromatin fibers take on a tighter, more solid form. The duplicated chromosomes now appear as two identical sister chromatids attached at the centromere. Mitotic spindles, which are composed of centrosomes and microtubules, also begin to take form. At the start of metaphase, the nuclear envelope disintegrates, and the microtubules enter the nuclear area and attach to the more condensed chromosomes. Further along in metaphase, the centrosomes make their way to the opposite sides of the cell, and the chromosomes align along the metaphase plate, preparing to be separated. At this point, all sister chromatids are connected to kinetochores on opposite poles. The cell will now enter anaphase. Anaphase begins when the proteins holding the sister chromatids are separated, meaning each chromatid is now a chromosome. The chromosomes begin moving to opposite ends of the cell, and as this is going on, the cell elongates. At the conclusion of anaphase, the opposite poles of the cell have the same number of chromosomes. In the final phase, telophase, daughter nuclei form in each cell, and the nuclear envelope begins to reappear, and the chromosomes become less condensed. In cytokinesis, the division of the cytoplasm is finished. In animal cells, a cleavage furrow using actin and myosin forms, pinching the cell into two separate, fully functioning cells. As shown in the clay, we started off the mitotic process with one cell containing 4 chromosomes. After cytokinesis, we end mitosis with 2 cells, each containing 4 chromosomes.
Contrary to mitosis, meiosis does not produce daughter cells that are genetically identical to their parent cell. Rather, meiosis is used to produce 4 gametes, such as sperm or egg cells, each genetically distinguishable from its parent cell. This allows for genetic variation in sexual reproduction. Mechanically speaking, mitosis and meiosis are very similar. However, meiosis has 2 steps: meiosis 1 and meiosis 2. Prior to meiosis, the late stage of interphase replicates a diploid parent cell's chromosomes, forming identical groupings known as sister chromatids. The cell is now ready for stage 1 of meiosis, prophase 1. In prophase 1, sister chromatids fuse with similar sister chromatids, forming homologous chromosomes. Crossing over, or the trading of genetic material, occurs between the homologous chromosomes at specific points known as chiasmata. Through this process, genetic variation will occur in the daughter cells. Also, in prophase, a spindle forms between the centrosomes. In metaphase 1, microtubules from the centrosomes attach to the centromere's kinetochores and line up along the metaphase plate. In anaphase 1, cohesion proteins are cleaved, and microtubules pull the homologous chromosomes apart. The sister chromatids, however, stay together. After telophase 1 and cytokinesis, 2 daughter cells form with a haploid set of chromosomes. In this example, that is from 4 to 2 double chromosomes. After meiosis 1, meiosis 2 progresses essentially like mitosis. Sister chromatids split. As shown in the animation, the 2 double chromosomes split in the daughter cells of meiosis 1 during anaphase 2, leaving 4 daughter cells of 2 single chromosomes each. The chromosomes are different from the parent cell. So how are mitosis and meiosis different? While mitosis and meiosis 2 practically follow the same mechanics, meiosis 1 is different because crossing over occurs in prophase 1 and homologous chromosomes, not single chromosomes, are paired and split with sister chromatids intact in metaphase 1 and anaphase 1. Additionally, mitosis results in 2 daughter cells identical to their parent cell, while meiosis results in 4 genetically unique cells.